Good morning or good afternoon, depending on where you are. So my name is Jerome Guicheux from the University of Nantes in France, and I'm a member of the Translational Research Committee of our lovely society, ICRS. So that is my pleasure to share this ICRS webinar dedicated to spatial omics and whether we can use spatial omics to decipher the role of immune cells in joint diseases. Spatial omics does allow us to map the distribution of mRNAs or proteins within compromised tissues, helping us to reveal how specific cell populations and molecular pathways contribute to inflammation. So we are very pleased to welcome two young, talented French scientists today with a very high level of expertise in spatial omics. So we're going to start with Dr. Soizic Garaud. Soizic is an intern, the French NIH researcher at the B lymphocyte autoimmunity and immunotherapies research lab in Brest, in Brittany, in France. She was a postdoc with Karen Vilat-Gallo in Brussels at the Jules Bordet Institute and Molecular Biology Lab. She's currently interested in exploring the immune mechanisms underlying immunotherapy side effects to develop predictive biomarkers and improve patients' treatment outcomes. Our second speaker will be Dr. Marie-Astrid Boutet. Marie is an intern researcher at the Regenerative Medicine and Skeleton Lab in Nantes, France, and she was a postdoc with Prof. Constantino Pizzalis at the Center for Experimental Medicine and Rheumatology at the Queen Mary University of London. And she was also a visiting fellow in the Department of Inflammation and Immunology in Milan, Italy, headed by Prof. Alberto Mantovani. Currently, Marie is interested in understanding how synovial tissues and notably its macrophage infiltrates can drive joint inflammation in the context of osteoarthritis. So for all the people connected, you can use the Q&A button if you have some questions, and I will be very pleased to read the questions after the second talk. So now, Soizic and Marie, the floor is yours. Soizic, maybe you can start. Thank you so much.
Okay. Hello, everyone. Thank you for the kind invitation to discuss today's spatial omics for addressing the role of immune cells in joint disease. And during this first presentation, I will give you an overview of spatial omics technology with example focus on rheumatoid arthritis. The next one. So just to remind you, rheumatoid arthritis is a chronic autoimmune disease characterized by joint inflammation and leading to cartilage and bone damage. So as you can see here, the infiltration is characterized by the presence of immune cells such as T cells, B cells, neutrophils, dendritic cells, and macrophages, but also non-immune cells, including fibroblasts. And all these cells produce pro-inflammatory cytokines, chemokines, but also enzymes. So next one, please. So to recent progress of the understanding of the rheumatoid arthritis disease have led to the development of new treatments such as biological therapy. So the first one is TNF inhibitors, including infliximab to block TNF, but also other biological agents. The first one is anakinra, an anti-IL-1, or tocilizumab, an anti-IL-6, abatacept to inhibit costimulatory signals, and finally the rituximab to deplete B cells. But of course, we still need to better understand the etiology of rheumatoid arthritis to improve patient outcome. So the next one. So to do that, next please. So the next one, to do that, we can analyze gene expression in joint. So we can use RNA sequencing technology. The first one, of course, is bulk RNA sequencing to identify gene differentially expressed. But here we have no information regarding the contribution of each cell. This limitation led to the development of single-cell RNA sequencing to analyze gene expression at single-cell level. Next. So here I would like to introduce one publication published last year in Nature, the work of Wang and colleagues. They characterize synovial cells from biopsy of patients with rheumatoid arthritis and osteoarthritis using single-cell RNA sequencing. So they analyze more than 300,000 cells, as you can see here, and identify some immune cells and stroma cells and other glial cells. And interestingly, here they subdivided tissue according to the cell type abundancy. And here they identify 6 different clusters of tissue. The first one was enriched in endothelial cells, fibroblasts, and myeloid cells. The second one in fibroblasts, the third one in T cells and fibroblasts. Group 4 was enriched in T and B cells, 5 in T cells and myeloid cells, and finally, the last group in myeloid cells. And what is interesting here is to observe the association of this cluster of tissue with treatment response. So here, for example, the presence of endothelial fibroblasts and myeloid cells were associated with a beta response to treatment. Next. And here it's another example of single cell approach. So here they characterize macrophages subsets using single cell RNA-seq, and here they identify 9 different clusters of macrophages. And as you can see here at the bottom, these clusters are sometimes enriched or decreased during the disease and in remission. Next. But here, the main limitation of single-cell RNA-seq is the fact that we have no information regarding the spatial information. Next. That's why the spatial RNA sequencing has emerged to combine the expression of gene expression with spatial information. Next. So there are two different techniques commercialized today. The first one is the 10x spatial transcriptomics, commercialized by 10x Genomics. Here they combine the power of microarray with barcoding using this Visium slide here illustrated. So these Visium slides have capture area with barcode. So here you can see two different workflows. In the first workflow, the first step is sample preparation, but here the tissue section is placed on the Visium slide directly, and then you can image to have the morphological context using H&E or immunofluorescence, and then the next step is hybridization, ligation, and barcoding. And after you can prepare your library and sequence. The second workflow is quite similar, but here the tissue sections are directly placed on a standard glass slides, and then you can image and hybridize your probe. And after that, we use the Visium cystasis to transfer a probe from the standard glass slides to the Visium slides. And the next step is probe extension, library construction, and sequencing. So using this approach, you can combine gene expression and spatial information. Next. The second technology is the Geomics Digital Spatial Profiler, commercialized by NanoString. So here the workflow is a little bit different. We use also barcoded probes, but here the tissue section is placed on standard glass slides, and you can combine with fluorescent antibodies up to 4. And here, using the Geomics, you can select regions of interest, and then barcoded probes are released with UV and collected before sequencing. And at the end, you can again see, you can analyze gene expression with spatial information. Next. But here there are some limitations. So the first one is the sensitivity of these techniques, but also the fact that we are not at single cell level resolutions. Next. That's why both companies have developed new instruments. So first, 10x Genomics has developed a new generation of slides called Visium HD slides, as you can see here on the left part. So with this slides, the capture area contain squares of 2 micrometers, enabling the single cell spatial resolution. Next. Not Visium, sorry. 10x Genomics have also launched a new instrument called Xenium Analyzer. So here the workflow is a little bit different. So the first step is always the tissue preparation. It's placed on the Visium slide. And then the second step is probe hybridization, ligation, and amplification. And after that, you can use the the Xenium Analyzer to image. So you can perform several cycles of imaging using fluorescence. And at the end, you can combine all images to see spatial information. Next. The second equipment is commercialized by NanoString and it's called Cosmic Spatial Molecular Imager. So here the workflow is a little bit different. It's based on fluorescent in situ fluorescent hybridization of fish, you can detect up to 1,000 RNA, and you can combine with protein. Next, just to illustrate that, I would like to introduce this publication of McDonald's and colleagues published this year in bioRxiv that characterize the synovial tissue of patients with rheumatoid arthritis, active or in remission, using the cosmics. And as you can see here, they were able to analyze more than 100,000 cells and identify different types of cells, so adipocytes, immune cells, endothelial cells. But interestingly here, they perform a neighborhood analysis to define what we call niches. So here, they were able to identify 8 different niches, and some of these were increase in active rheumatoid arthritis, such as myeloid-rich lining niche, number 2 in orange, or plasma-rich niche, number 7 in pink, and lymphoid niche in blue, compared to remissions. Next one. But of course, the next step is to move to proteomics. And again, here you have several techniques. Next. So first, what we can do is multiplex histology. And I would like to introduce here the PhenoImager commercialized by Akoya Bioscience. So here it's very simple. It's based on immunohistochemistry. So the first step is, of course, the incubation of primary antibody, and then secondary antibody conjugated to HRP. And here the signal, the fluorescent signal, is then amplified using pyramid. And at the end, we can remove primary and secondary antibody and repeat several cycles of staining. So here we can do up to 6 markers in a single session, and at the end, all fluorescents are applied using the PhenoImager or Polaris. It's the same. Next. Another approach is high-plex immunofluorescence using the Orion machine commercialized by Raresight. So here it's a little bit different. We use fluorescent antibodies. It's a one-shot staining. So it's really simple, and everything is required with the Orion. So here we can do up to 16 markers at the same time, plus DAPI and some channel for autofluorescence. Next. And I would like to illustrate this approach with one of our work here in Brest. It's a retrospective clinical trial to characterize inflammatory arthritis induced by immune checkpoint inhibitors in cancer patients. So here we collect biopsy from patient, and we compare with rheumatoid arthritis using the Orion equipment. So as you can see here in these two representative example, we observe the infiltration of immune cells, so T cells in blue and green in both cases, but also B cells here in red. And what we can see in ICI-induced inflammatory arthritis is the presence of proliferative B cells, not observed in rheumatoid arthritis. So this work is ongoing in our lab to better understand the contribution of B cells in the pathogenesis of ICI-induced inflammatory arthritis. Next one. But of course, the main limitation is the number of markers. So another approach is mass-based imaging. So here you can analyze up to 40 markers simultaneously using antibodies conjugated with metals. So, and in both cases, these metals are detected and quantified using a time-of-flight mass spectrometer. So there are 2 different technologies commercialized today. The first one is MIBI technology from Beyond Path, and the second one is imaging mass cytometry using the Hyperion commercialized by Standard BioTools. And I will give you one example later. Next one. And the last approach is DNA barcoding techniques. So it's commercialized by Akoya Bioscience. It's the PhenoCycler or Codex. So here it's a little bit different because we use the antibodies conjugated to barcode. And after that, we perform again several cycles of imaging with fluorescence. And at the end, we combine all images. And here we can visualize 20, 40, or 100 protein in a single session. Next. So just to introduce the workflow for images, so it's always the same. We can use several tools. So we have we have software like Halo, QPath, and VisuFarm, but we can also use R tools. And it's always the same. So the first step is the tissue segmentation. If you want to compartmentalize, for example, lining, sublining in rheumatoid arthritis. The second step is cell segmentation. So here we use marker of DNA to identify each cells. And after that, you can do phenotyping. And the final step is spatial analysis. And in some cases, you can do 3D organization or spatial temporal kinetics. Next. So to give you one example, I would like of imaging mass cytometry, I would like to introduce the work of Marie-Astrid and her colleagues conducted at Hermes. So this paper published last year in bioRxiv. So they use imaging mass cytometry to characterize macrophages in the synovial tissue of patients with rheumatoid arthritis. So here they use a panel of 33 antibodies to target macrophages, so resident macrophages and inflammatory macrophages, but also lymphoid cells, so T cells, B cells, but also stromal cells and some marker of activation. And they analyze healthy tissue, but also tissue from patients with osteoarthritis, polyarthritis, active and in remission. So here you can see some representative example with the filtration of macrophages in purple, lymphoid cells in blue, and stromal cells in green. And on the right part, they were able to identify 17 subsets of cells. So you can see several subsets of inflammatory macrophages, for example. Next. But interestingly, what I observe here in this publication is some alteration in the density of some subsets. So here in active rheumatoid arthritis, they observe decrease of resident macrophages in blue compared to healthy tissue and osteoarthritis, associated with an increase of lymphoid cells, including T cells and B cells in brown, and also inflammatory macrophages here in green. Next. And they also perform neighborhood analysis and identify several niches, in total 9. So 3 in the lining and 6 in the sublining. And as you can see on the right part, this is the composition of each neighborhood. So L1, L2, and L3 are in the lining, and we can see enrichment in resident macrophages. Type 1 or type 2, but also fibroblasts. And they demonstrated some alteration in active rheumatoid arthritis. It's on the right. So with, for example, a decrease of L1 associated with an increase of L3. And interestingly, they demonstrated that DMARD's treatment with methotrexate can restore homeostasis with an increase of L1 and a decrease of L3. So next. So just to conclude this presentation, so of course Spatial Omics is very interesting to analyze gene expression of proteins at single cell level. We have a lot of information, but of course there are some limitations, and the first one is the cost. These techniques are really expensive, and also the complexity. It's sometimes really challenging. Another limitation is a sample preparation, especially if you work with bone or cartilage. You have to prepare the tissue, and this can impact the quality and the viability of cells. And finally, the last challenge is also the analysis. So it's highly complex, and you need some tools and maybe some bioinformation to integrate all this information and data. So thank you. Next. So thank you for your attention, and I'll be happy to take your question after the presentation of Marie-Astrid. Thank you.
Thank you so much, Soizic, for the very, very nicely illustrating presentations. Don't forget to write your question using your Q&A button on the system. So now, Marie, the floor is yours. Maybe you can start your presentation.
Yes.
Thank you again,
Sure.
Soizic.
Can you see my screen? Is it okay?
Yes, perfect.
Okay, perfect. Thank you so much. Thank you for the introduction as well and for the invitation. So the idea today, following the first presentation that was more technical, is to give you an example of how to use spatial omics in the context of the study of joint diseases and, in my case, osteoarthritis. So as you know, the joint is composed of many tissue that can all be affected by osteoarthritis. Osteoarthritis has different inflammatory and degenerative features, such as you can observe synovitis, so the inflammation of the synovial tissue, cartilage degradation, for example, or the presence of osteophytes. So this is a very complex and heterogeneous disease of the whole joints. Osteoarthritis affects over 50% of the population age of more than 65 years old, and affects over 500 million people worldwide. The etiology of the disease is still very unclear, with various risk factors that we are still trying to understand. And at this stage, there are only symptomatic treatments, so no strategy able to diminish the progression of the disease or to stop its progression. So as I said, this is a very complex disease with distinct phenotypes that have been observed and described in the literature, but also different underlying molecular endotypes that have also been described but are still poorly understood. So in our group here, we focus mainly on the synovial tissue, and this tissue in the healthy or physiological context is composed of two major parts. So the lining part of the synovial tissue is composed mainly of macrophages and fibroblasts, while the sublining is a connective tissue composed also of fibroblasts and macrophages. You can see some vessels as well. And in the context of osteoarthritis, what we can observe is that this lining tissue gets thickened with up to 10 to 20 layers of cells sometimes. And in the sublining tissue, we can observe a lot of infiltration of distinct immune cells, such as macrophages, B cells, or T cells, sometimes forming aggregates, and also some neovascularization. So at the moment, the gold standard to score the synovial inflammation in osteoarthritis, but also in other joint diseases, inflammatory joint diseases, is the Kranz scoring system. So this system is actually composed of three different criteria. One is looking at the thickening of the lining layer, as I told you, but the other one is the density of the synovial stroma, and we also score the importance of the inflammatory infiltrate and mainly the lymphocytes. So this score has been used in many joint disease and is very well accepted in the community. But as you can see, we really have in the context of OA here a lot of variability, and this Kranz scoring system is also not really well characterized to be associated with specific clinical features. So this is complicated to make links with the disease development to study synovitis. And so just a parallel with what has been done in the past in the context of rheumatoid arthritis. So the group of Professor Pizzalis in London has described different subtypes of rheumatoid arthritis based on the histopathology. And as you can see here, they describe three main subgroups of patients based on the infiltration of distinct immune cells, plasma cells, macrophages, B cells, and T cells. And these subgroups of synovial tissue have been actually shown to be characterized by a specific transcriptomic signatures, but also to associate with specific clinical features and to predict the response to treatment in rheumatoid arthritis. So this is a characterization that has been super interesting to drive the development of personalized therapy for RA. In the context of OA, what we have shown this year is actually that we can find the similar pathotypes. So just to give you a little bit more of details, the post-immune pathotype is characterized mainly by the presence of some macrophages. This is a post-immune tissue with really low level of inflammation and really low amount of immune cells. In the case of the diffuse myeloid pathotypes, we can observe the main infiltrate of macrophages in the lining, but also in the sublining, and some T cells, sometimes B cells, are scattered all around the tissue. And this last group of tissue is actually quite interesting for us, as we can observe in the lymphomyeloid pathotype some aggregates of T cells and B cells that are surrounded by plasma cells, but also these tissue are really infiltrated by lots of macrophages. So what we have shown is actually that compared to what's happening in RA, here we are at late stage of the disease, the percentage of the lymphomyeloid subgroup pathotypes is actually quite high, and this is what quite surprising for a disease that has long been considered as non-inflammatory. So we are very interested to understand better what's happening here. And what's is of specific interest for us is that this scoring system, so the pathotype scoring system, is really non-redundant with what's happening when we do the CRAN scoring system, as for example in the lymphomyeloid, you can find some synovial tissue characterized by a high CRAN score, but also a low CRAN score. So this is really a new view of the categorization of synovial inflammation in a way. And importantly, what we have published in this paper in 2024 is that this the presence of each pathotype associates with distinct level of peripheral inflammation, here represented, for example, by the level of lymphocytes in the blood, but also with some monocytes to lymphocyte ratio or neutrophil to lymphocyte ratio that has been associated in the literature to associate with also the severity of the disease, the radiographic severity of the disease. So in here, I'd like to highlight, because this is important for the rest of my talk, that the macrophages are actually present in all the pathotypes, but what we are really trying to investigate at the moment is that is there like a difference in the phenotype of these macrophages, and obviously the communication of the macrophages with the other cells surrounding in the synovial tissue. So the idea here of this talk is actually to highlight two of our main objectives here in the group, and to give you some example of how we use the spatial omics for addressing our question. So one of our main objectives is to characterize the synovial tissue heterogeneity, to define a relevant stratification strategy for OA patients. And here we really want to use unbiased approaches, and also in the context of spatial characterization. And our second objective is actually to look at how to target synovial macrophages for the personalized management of OA. And here in that case, we will have really a targeted approach with some panels that are designed to look at macrophages and their neighbors. So to start with, what we did following the publication of our first paper was to develop an independent cohort of OA tissue at North University Hospital to look at the features of synovial tissue. So just briefly, following joint surgery, joint replacement surgery, we collect synovial tissue, adipose tissue, and cartilage, and perform all the histology and scoring. And at the moment, we collected over 150 samples. So just briefly, the results here of the first cohort is the one I showed you that we published this year. And these are the results of our independent cohort. So you can still see that there is a majority of lymphomyeloid tissue in this cohort. And so meaning that most of them are characterized by the presence of ectopic lymphoid structures, so these aggregates of lymphocytes. Here again, we can find that there is no redundancy with what is observed with the CRAN scoring system. And importantly, the presence of each pathotype doesn't correlate with the age of the patient. So this means it's not because we are aged that we have a fibroid pathotype, or also with the disease duration. So this appears to not be linked with the disease duration. So what we are trying to answer here is, are histopathotypes associated with specific transcriptomic signatures? So our preliminary data indicate that yes. And so what we did was to perform some bulk RNA sequencing, so at the level of the full synovial tissue. And what we can observe from this graph on the left is that there are some specific transcriptomic signature that seems to be associated with the level of tissue inflammation and the immune cell infiltration. So at the moment, what we are focusing on is to better identify the pathogenic pathways that are related to osteoarthritis in order to better define the disease endotype, as I told you in introduction. And for that, we are analyzing at the moment a very large cohort in collaboration with the Center for Experimental Medicine and Rheumatology in London, with 8 post-immune patients, 39 diffuse myeloid and 45 lymphomyeloid patients to complete and compare the data with what we observed here on a lower amount of tissues. And so now to move on to the spatial part. So as I said, this first part has really as an aim to characterize the tissue with an unbiased view. So what we are looking at is to see whether histopathotypes are also associated with some niche-specific proteomic and transcriptomic signatures. So as I told you, the different parts that are of interest for us are the aggregates, the lymphoid aggregates, but also the lining and the sublining. So for that, we used the geomics experiment from nanostring. So that has been already described in the previous presentation, but just very briefly, we are looking both at the protein, but also at the RNA in some region of interest. And then the oligos that we use, the different probes are cleaved in the region of interest, and then we do analyze with the sequencer. So in our case, as I told you, we use some morphology markers to really observe our region of interest. In this case, CD3 and CD20 to look at the aggregates, but also CD68 to differentiate from the lining and the sublining of the tissue. And this is a strategy that we have also published in a previous paper in the context of rheumatoid arthritis in Nature Communication. For this study, we selected some panels. So for the protein panels, we looked at immune cell profiling, but also activation and cell typing. And we combined this analysis to a whole transcriptome analysis using the geomics. In this case, we have 16 OA and 4 non-OA controls, as well as 4 array controls that have been selected to also present some aggregates and be a characteristic of what we can observe in array with the presence of autoantibodies. So as you can see from this graph, actually the unsupervised clustering is really showing us that the 3 different regions of interest are really co-localizing together in this clustering, with here the ectopic lymphoid structures, the lining and the sublining of the tissue, which is also reflected here on this graph with the PCA of the ELS lining and sublining having at the protein level here really a nice segregation. And using this technical approach, we can also validate the expression of some protein of interest. Here I put as an example CD45 that you can see is really highly expressed in the ectopic lymphoid structure, so the lymphoid aggregates, mostly here in the lymphomyeloid and the array, but and that the expression of CD45 is quite similar in lining and sublining. So this is quite powerful because here we are accessing to protein expression, and in our case it was really to confirm that our structures were well characterized. But of course what's it's even more of interest for us is the RNA expression, and in that case, so we are really at the beginning of the analysis, but I just wanted to show you this graph that is also confirming that the profile of ELS, lining and sublining are really distinct at the gene transcriptomic level, and at the moment we are focusing really on identifying disease and also pathotype specific signatures within each synovial niche, so ELS, sublining and lining. So this was for the unbiased approach, let's say. So the second example I'd like to share with you is about macrophages, because we really focus our research on looking at the distinct profile of macrophages in the synovial tissue of OA and designing some personalized tools to target them in the synovial tissue. So just as a brief reminder, macrophages are really present in all the pathotypes that we observed in osteoarthritis, and macrophages have been shown already in the literature to be like really central player in the synovial tissue of osteoarthritis, as it actually produces lots of factors that influence what's happening in the synovial cavity and on the cartilage, for example, but also they are able to attract more inflammatory cells to the tissue, among other functions, of course. So as Soizic showed in the previous presentation, there are actually lots of studies in the context of rheumatoid arthritis that focused on the diversity of macrophage phenotypes. So as you can see here, this was one of the first studies showing in array synovial tissue that there were 9 different clusters of macrophages present in the synovial tissue. And here more recently in Nature, a really nice paper that Soizic presented before, showing also that we can really find a large diversity of this type of cells. So in parallel to what I showed you in the first part, what we are collecting is also a cryopreserved sample of synovial tissue when we collect the rest of the tissue. And from this synovial tissue, we perform single-cell RNA sequencing of all the immune component of the synovial tissue. So we were able to find lots of macrophages as expected, but also T cells, B cells. Here are just a few cells that passed the sorting of CD45, so some fibroblasts left over. And then we subset again using only the macrophages to look at the distinct population of macrophages in the synovial tissue. We were able to find 13 myeloid cell population that are all characterized, as you can see from this graph, by specific transcriptomic signatures. And interestingly, these 13 population are actually differently represented across pathotype, and we identified some population of interest here, the third one, fifth, and eighth one, that are more present in some of the pathotypes. And we are now working on targeting specifically this population in each of the pathotypes. But as Soizic highlighted before, here we have the problem of the tissue dissociation, because for running all these single-cell RNA sequencing experiment, we have to dissociate the tissue. And in this process, we may lose some cells that are more fragile. And it's really important to focus on what's happening in the tissue, with the localization of each cell. So we are focusing now, obviously, on confirming all these data on by immunofluorescence on the tissue, but obviously, as this is the topic today, also using spatial omics to address our question of how the macrophage behave in the tissue and with which cells they communicate in the tissue. So these are the main questions: how they organize and how they communicate. So for this, we use a second technical approach that is this time not unbiased, but really biased, because we are looking at specific panel of genes. And in our case, we really design a panel that is looking at macrophages. So I'm not going to go into too much detail for the Xenium, because Soizic already introduced the technical approach very well. So here we actually don't have to select any ROI, as in the first part when we looked at the aggregates or the lining, sublining. Here we will analyze everything that is in the sample area on the 10x slide. Then, as I just mentioned, we looked at specific panel of genes. So in our case, we use multi-tissue and cancer panel. That is a large panel, very well designed to look at immune cells. And also we added a custom panel of 100 of genes that we selected based on our previous results on the single-cell RNA sequencing experiment. And in this case, we selected 4 OA tissue, 3 non-OA controls, and 3 array that are the same as I showed you for the geomics. So we will be able also to compare both technical approaches. So here, really importantly, what I wanted to highlight is that we were able to validate our single-cell RNA sequencing data, both from the clustering part, but also from the proportion part. So here I'm showing you how the UMAP looks like in the different tissue that we selected, control, the 3 pathotype of OA, and the array. And importantly here, we really have access to some information on top of what we had with the single-cell experiment. For example, here the yellow population is the adipocyte population that usually we lose in the single cell experiments because they are very fragile. And this is super interesting to have them and to be able to observe them also in the tissue. This is just one example, but here we really have a very nice view of all the cell type present on the slide. And here, just a few images to give the context. We are able to visualize the cell cluster, of course. So here on this image, each cluster is represented by one color, and you can see that very nicely. We are able to look at the lining, but also the vessels and the cells that are around the vessels. So we really have now the spatial organization of the tissue. And if we want, we can select one gene of interest. Here I put PRG4, that is a fibroblast, one of the fibroblast marker. And you can see that this is really nicely expressed in the lining as we expect. So at the moment, we are working on spatial niches, as Soizic mentioned, and also looking at the interaction of each cell type with their neighbors. So stay tuned for more, it's coming. So just as a brief conclusion, as I hope I demonstrated here, spatial biology will really help to decipher the organization, the relationships, and the functions of the cells within joint tissue. And here, in our different project, what we really aim to do is, thanks to spatial omics, better define the molecular endotypes of osteoarthritis, obviously, at the end, to be able to design some targeted and personalized treatment. So with that, I'd like to thank you for your attention, of course, but also all the funding agency, my group here at Hermes in Nantes, especially Nicolas, Anaïs, and Julien, who participated to generate this data, our collaborators in clinic, our collaborators abroad, but also the different facilities that help doing all this experiment. Thank you very much.
Thank you so much, Marie, for the very nicely illustrating presentations. Now I think it's time to move to the Q&A session. We only have at the moment only one question in the chat, but so I'm gonna read it, and I think this is for you, Marie. The question is coming from Dominica Odenschild, and the question is: how variable is the composition of synovial tissues in an OA knee joint? And does the biopsy site greatly affect the observed histopathotype?
Okay, yeah, so that's a very important question, actually. So what we were able to do regarding the joints is to compare the hip OA and the knee OA in the paper we published, and we observed that actually the proportion of each pathotype was very similar in hip and knee. So at the moment, we only focus on both of them. And that's why we also decided for the rest of the experiment to focus only on the knee at the moment. And we will be able in the future to go further and to also look at other joints. And according to the distribution within the tissue, what we always do now is to, when we collect the tissue, to look at different section and the different part of the tissue to really cover the whole joints. And we, like in most of the case, and that's what also we demonstrated in the first paper, we are able to really find similar pathotypes across all joint.
Thank you, Marie. So we have another questions from Alexander Huber. And the question is, how would you predict how pathotype profiling would be used clinically to identify the potential treatment options?
Okay,
I think maybe you can address the question, maybe Soizic and Marie.
Yeah, I can start if you want, Soizic. So really The idea here is, so at the moment, the data we are showing are at late stage of the disease. So at the moment, we are in the process of looking earlier in the disease using synovial biopsies. And my opinion on that is really that if we are able to confirm early in the disease development the pathotype of the patient, we could be able, thanks to all the analysis we are doing at the moment, be able to define some pathogenic pathways that are related to each pathotype and better attribute the treatment option. So this is obviously a very long work and a long path to arrive to personalized treatment in a way, but we are really hopeful that this kind of strategy could lead to better addressing the treatment for each patient.
Thank you, Marie. If you want to comment, Soizic, you're also welcome. But I have a question for you, Soizic. We have seen the complexity of all the technologies you have described. I just want to know what is the most expected evolution in spatial proteomics, because we see that there is still a limited number of targets that we can address currently. What will be the next evolution in spatial proteomics?
It's a good question. I really don't know. For me, the main limitation for the moment is how to analyze all this data. And of course, we can have more markers and more targets. And the main limitation will be how to analyze and integrate all this information. So I don't know what will be the future. We have a lot of tools for the moment. It's a huge revolution this last 5 years, I would say. So we will see. We will see. I don't know.
Okay. So we have another question for you, Mary, coming from Lindsay Lamlin. The question is, have you identified any associations between the histological endotypes and clinical pain, specifically pain? This is a very interesting question. And the rest of the question is, are there pain-relevant gene or protein markers overexpressed in this?
Okay, unfortunately, so yeah, this is a very important point because we know pain is really one of the major feature of OA patients, and sometimes unrelated to what's really happening in the joints. So at the moment, no. The answer is no. We didn't look at the relationship between histopathotypes and pain. We actually focus almost only at the moment on late stage disease. So they are patients that are undergoing joint replacement. They are almost all painful. But what we are trying to do at the moment is building this new biocollection of OA patients at earlier stage. And in this one, we will have a better characterization of the clinical features, including the pain. And this is definitely a very, very important point to address in the coming months and years. And so for the second part of the question, actually pain-relevant gene protein markers, I cannot answer this question at the moment, but this will be something to address if we are able to see a specific signatures that is associated to pain. And this has been done in other contexts, such as, for example, the composition of the microbiota or other large-scale studies. So I'm quite hopeful we'll be able to also do the same in the synovial tissue and related to histopathotypes.
Okay, thank you so much. Very interesting. For you, Soizic, we have questions coming in the chat, so maybe I can start with these questions coming from Adi Lakush. I think it's for I don't know, Mary or Soizic, I don't know. Did you detect any sexual dimorphism in the expression of inflammatory cells or mechanotransduction markers, or is it? Did you have a look on the is there any difference in terms of sex in your experiments, and how can you use these expensive technologies in patients? How do you see the future of anatomopathologies at the hospital, for example, because this is costly technologies. So for the first part, did you detect any sexual dimorphism, Marie, in your experiments, or?
So at the moment, we checked the distribution of the pathotypes according to age, and we see absolutely sex. we see absolutely no difference between men and women. So yeah, that's the only key, only answer that I can provide at the moment. Regarding transduction marker, I actually didn't check. So, but this is an important point to keep in mind for the futures. I don't know, Soizic, in RA or in other arthritis things you know. Okay,
and concerning the cost, and because this is very, very expensive technologies, how do you see the transfer of those concepts from the bench to the hospitals, for example, for diagnostics or?
Yeah, that's true. So cosmics and geomics, for example, is really, really expensive, but the goal after is to really identify some markers. So we can imagine to identify 1 or 2 or 10 markers and then use cheaper technologies such as immunohistochemistry and to perform some testing routine. So here, for example, in pathology lab, we have more and more IHC to detect inflammatory cells in tissue sections. So, and the future is also the numerization of the pathology department. It's already going here in France. So to numerize all the staining and then use deep learning, for example, or some artificial intelligence to stratify patients and better identify patients for treatment.
I agree with you, Soizic, that definitely looking at smaller panel would probably help reducing the cost, and definitely these technical approaches, we can apply them to really refine the panel for going to clinical application. One thing also that is really inspired by what has been done in the field of cancerology is the development of TMA for preparing the tissue. So because all these technical approaches are on tissues, fresh or FFP tissue, but on FFP tissue in the cancer field, they use a lot the tissue microarray to be able to collect some tissue and put everything in one slide to be able to analyze in the small portion, small area of the slides as we showed a lot of different tissue. This is possible, and this I think in rheumatology should really be implemented in the future, at least for the study of the synovial tissue. This has more challenges, of course, for bone and cartilage, but in the field of synovial tissue, I really hope we can get there.
Okay, and because I know that you are in the process of purchasing specific equipment in Nantes, Marie, can you give us an idea about the price of those technologies regarding the equipment, and also regarding how many euros we need for very simple experiments with a very limited number of samples? But maybe you can share your experiments in that.
Yeah, so actually the equipment in themselves, they are, I mean, not so expensive if we compare to other kind of equipment for lots of experimental settings. So it's, I mean, really depending on the approach, because Soizic presented a large panel of equipment, but I would say between 300,000 and 500,000, let's say, something like that, just for buying one equipment, depending on what you want to do. You want to have a precise idea of if you want to go for unbiased characterization or more biased, so go for more geomics and Visium, or on the other side, Xenium or other technologies. Really regarding the experiment, it's really variable depending on the number of sample you have, because the more you have, the less the panel costs, and the size of the panel, what you want to do. Just to give you one example, but I don't know if it's going to be representative of everything you can do, but for a Xenium experiment using, for example, 4 slides, so 2 batches of Xenium experiment with like a 500 gene panel that has been made custom for one experiment, it's about 20 kilo euros or something like that. So it gives you a rough idea of the cost you should plan for this kind of experiment, but this is really dependent on your settings and questions.
Okay, thank you so much, Marie. We have a last questions coming from Ellen McCarthy regarding your presentations. You have both expressed how challenging this technology can be for assessing bone or cartilage, and just how difficult it is. Do you recommend FFPE or FF, depending on probably on the experiments you want to do? What other samples prep will you require?
I think FFP is a good option. The only thing by talking to the different facilities or even the people who are selling the machine is that they didn't do a lot of tests at the moment on cartilage and joints. So some of them start to do the preparation. I think the key point here is really the fixation of the tissue and the preparation in case of decalcification, for example, to really try to have a condition that will not be too harsh on the tissue and on the RNA, because we really want to keep the RNA safe and on good condition to be able to perform all this experiment. So I don't have like a precise answer to provide because no one has, I think, to my knowledge. I didn't see really lots of publication using this high-plex spatial experiment on cartilage and bone. But yeah, definitely we can keep in touch for this kind of question because I'll be very interested to optimize this in the future.
Okay, thank you. Soizic, if you want to comment regarding FFPE or freshly frozen or something like that, that's okay. We have another questions, and we still have 4 minutes before the end of this webinar. This question is: how can spatial omic techniques such as IMC, imaging mass cytometry using metal tagged antibodies, be applied to better understand the role of immune cells in joint diseases? Could these approaches, in combination with polymeric biomaterials, enhance diagnostics or therapeutic strategies by providing detailed spatial maps of immune cells interaction within the joint tissues? This is a very difficult question, but so what about the possibility of combining such kind of approaches with polymeric biomaterials to enhance the diagnostics? What is your opinion regarding this? Is a very difficult question.
Still trying to read it to understand everything. So I think if you want to start on the first part on imaging mass cytometry, that's fine. Yeah. Sorry, it's a long question.
Yes, yes. You still have 2 minutes to read it. Sorry, Sarah, but this is a very difficult question.
Yeah, I don't know. Maybe Marie, you have an answer.
No, this is just the part on the polymeric biomaterials. I'm not sure I really get the point of the question.
I think this is if I correctly capture the questions, this is to combine detection of specific population of cells with targeted delivery of therapeutic strategy using polymeric biomaterials. So maybe you can do that at the same time.
Well,
I don't know. I don't know if.
As long as the tissue that has been observed with lots of, I mean, everything that you want to test on this tissue, you're able to fix it and then cut it for further spatial experiments. I don't see a problem to try it, but with biomaterials, I guess we'll have same problems also on the preparation of the samples. So this will be really project specific. And probably not easy to have a general answer on that. But yeah, that is part of the challenge. I don't know if you mentioned this, Soizic, on the last slide, part of the challenge that is a very new technological approaches. There are lots of possibilities on the market to different equipment, but they all are very complementary and all address very specific questions. So yeah, we need to progress on our understanding of how to prepare all type of samples. Sorry, it was a very general answer, but I don't have more precise.
So we're gonna record the questions because we still have questions coming in the chat. Don't have time enough to address all the questions we have, so we can have a look on that after. I want to thank our 2 speakers, and I want to thank the audience also for the large number of questions we received. This is a very, very interesting topics, and do not hesitate to be in touch for if you have specific questions with Marie, with Soizic, or with me. And we're going to be very, very happy to address all the questions you have. So thank you so much for joining us today. And it's time to close this webinar. Bye-bye.
Thank you very much, everyone.